My name's Stephen Atkinson. I'm a grad student at Oregon State University, although you can probably tell by my accent I'm actually shared between Oregon State University and the University of Queensland in Australia. I started my work on the Klamath by focusing on two sites, one in the upper basin and one in the lower. We know from Sasha's work that the Ceratomyxa Shasta levels in water are the same at both sites. And so the question is, what happens when we expose two different cages of fish at these locations? How do the fish respond to the parasite? So we used cages of out-of-basin susceptible rainbow trout and Chinook salmon exposed at the same time at both locations in the river. If we put a cage of susceptible rainbow trout in the river, if there's any levels of spores in the water, they will all die. The other group of fish we used were Klamath River Chinook salmon. These are a species of interest, species of concern in the basin, and we source them from Iron Gate hatchery. So these are fish that have had long-term exposure to the parasite and they have a certain level of susceptibility. If we take a cage of the Klamath Basin Chinook salmon, expose them in the river, you'll find that depending on the level of spores in the river, either sort of up to 50%, 60% of them will die. They won't all die. They're susceptible, but they have a certain tolerance. We then took them back to the lab and recorded what happened to the fish. So what you see here are two graphs representing the upper river basin results and the lower river basin for the rainbows and the Chinook. You can see two main differences between these groups. First of all rainbow trout died no matter where we exposed them. We expected to see this as we knew these out-of-basin rainbow trout are very sensitive to Ceratomyxa Shasta compared with the native Chinook. And secondly, what we didn't expect to see was that there was a difference between the number of Chinook that died in the upper and lower basin. A lot more died here in the lower basin. So the question is, why is this so? Are we seeing a difference here in the parasite between the upper and lower basin? So we had two questions from these infection results. Is the same parasite present? Is it the same species in the upper and lower river basin? And if it is the same species, are we actually seeing some sort of strain effect, much like you would with a different sort of strain of the flu virus? Are we seeing a different strain in the lower basin versus the upper basin? So to answer the question of whether we had two different species or whether we had just different strains of the parasite between the upper and lower river basin, we turned to DNA sequencing to find out. Sequencing a gene that we know and love from this group of parasites. It's been very useful to us, the 18S gene. The 18S is an interesting gene in that, unlike most genes which are DNA, they get converted into protein which builds your body. The 18S is a ribosomal gene. So what it is, it sits in the nucleus like most of the other DNA, but it gets converted directly into RNA, which is used to build the structure of a ribosome. The gene, we know from previous work with the myxozoans, this group of parasites that includes Ceratomyxa Shasta, we know that the 18S is a good gene for looking at where you get very little variation in members of a species, just samples from the same species from different locations, for example. But when you look at different species of myxozoans, you'll see on the order of sort of 2% differences. So this gene has got like 1,700 of these nucleotide letters in it. And of those, you get about 10, 20 differences in a gene typically, and that's enough to say, right, this is species A and this is species B. We knew that if we sampled parasites from both the upper and lower river basin, and the 18S gene was quite different, that we'd be dealing with different species. So that's exactly what we did. We took some upper river samples, some lower river samples, and what did we find out? The short answer is that there isn't much variation between the upper and lower river basin. Of the 1,700 nucleotide letters that make up this gene, there were only 3 positions where we found differences. And this, in terms of are these different species, this is quite a conclusive no. This level of variation is quite typical of what we would see with different samples of the same species from different regions.
Research question 2. Are we seeing different strains of C. Shasta?
But still, the fish data suggests that there were different parasite strains. So we needed to sequence a more variable region to get this information. And really quite handily for Ceratomyxa Shasta, right next door to the 18S is, it's called the internal transcribed spacer region. It's ITS for short. It's not a gene. As far as we know, it doesn't actually do anything in the finished organism. But the advantage of the ITS is it's known from other parasites that it has a lot of variation. It usually has too much variation to be useful when you're distinguishing species. But we thought it might be a good target for Ceratomyxa Shasta because it would have more variation than the 18S. The next step was, like we did with the 18S, we sequenced a range of samples both from the upper and lower river, from the different fish. The initial study was done using rainbow trout and Chinook salmon, which we had exposed both at one site right above the dams in the Williamson River, come Klamath Lake, and the second site was below Iron Gate Dam in the lower river.
You will remember that Sasha's work showed that Beaver Creek and the Williamson River Delta had the same amounts of sea Shasta parasites in the water.
So I sequenced the DNA from the parasite from these different fish species, looked at the ITS, and found that indeed there was quite a range of variation in the ITS. We found 10 times more variation than we did in the 18S.
So, for the 1700 nucleotide 18S gene, there were 3 nucleotide positions that varied. However, for the 200 nucleotide ITS, there were 20 nucleotide positions that varied.
Secondly, what we realised very quickly was that there was one particular spot in the ITS that had 3 letters in a row, ATC. And what we saw was that depending on the sample we got, it either had none or 1 or 2 or 3 repeats of this. So it was either ATC or ATC, 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 ATC. And it was a very handy way of just looking at a sequence and going, right, this has got no repeats, this has got 1, this has got 2. And that's where we call our genotypes from. Genetic type 0, genetic type 1, genetic type 2, and so on.
Research question number 3. Is there a relationship between the sea Shasta strains and the different salmonid hosts?
We were very excited to find that the samples split up quite nicely according to the fish host and according to the location. We noticed very early on that all of these out-of-basin rainbow trout that we'd exposed, no matter where we exposed them, they caught the type 2 strain. Presumably they've never been exposed to Ceratomyxus shasta. In the upper river and lower river they were the same. The rainbow trout were genotype 2, whereas the Klamath Basin Chinook salmon were different depending on where we exposed them. We knew from the infection data that very few of the Chinook got actually infected up in this upper river site. Of those that did, we saw a mixture of genotype 2 and genotype 3, but very much a low-level infection because these fish weren't dying in the upper river. When we moved to the lower river side, a lot more of the Chinook salmon had died, and when we did the genotyping of them, we found that they were entirely genotype 1, which was a striking contrast. The rainbow trout right beside them had genotype 2, whereas the Chinook salmon had type 1. And these are fish exposed at the same time in the same water, and yet there's a very strong sort of strain-specific response from the different host species. Now, I think the key to that is when you look at the water samples. So in parallel we had the water sampling, which Sasha had done, and we knew from that that the parasite levels were the same in both the upper river and the lower river. But then when I did the ITS sequencing and I genotyped these samples, what we found was that there was quite a different mix of genotypes in the water. In the upper basin, Ceratomyxus shasta had type 0 and type 2 in the water, whereas in the lower basin it had type 2 again, but it had type 1. We didn't actually see any type 0 in our initial study. So we know that Chinook salmon get type 1 and the out-of-basin rainbow trout get type 2, but what fish species do the other genotypes infect? To wrap up my part of this study, I did some more DNA sequencing of Ceratomyxus shasta samples from 3 other salmonid species important in the Klamath Basin: coho salmon, redband and steelhead trout. The redband trout and the steelhead, they're all the same species, as well as the susceptible rainbow trout. They're all Oncorhynchus micus, the one species. I've summarised the data in this table. The type 0 was found at all sites, upper, middle and lower river sites. However, it was only hosts with steelhead and redband rainbow trout. This type 0 isn't usually causing mortality in them. There's a tolerance. We get, I think, anywhere from 10 to maybe 50% mortality at the most. But in general, the mortality is very low in steelhead and native redband rainbow trout. The type 1 was only found in the lower basin, and its host was Chinook salmon. Type 2 was found in both the upper and lower river basin, and we think its natural host in the system is the coho salmon. We know these susceptible out-of-basin rainbows will catch it regardless, but it was in the native coho that we saw a strong signal for it. Type 3 we found in fish in the upper and lower river basin. We never found it in water samples, which suggested to us that it was only ever present at a low level. However, it was found in all fish species, which suggests that type 3 is quite a general strain of the parasite.
Discoveries of different strains of sea Shasta and their relationships to different salmonid species raise new challenges. For example, Sasha wants to develop an efficient molecular method that distinguishes different strains in the water. Stephen and Sasha are looking at different strains and polychaete infection. Adam will be investigating the proportion of different strains in his epidemiological model. The lab is also interested in exploring the evolutionary progression of the different parasite strains and the different salmonids.